Okay, so welcome to this first thematic session. My name is Asla Pitkänen and I'm chairing this together with Michele Simonato. So before we continue with the session, thank you very much, Dr. Berkuk, for this information. Related to that, I would like to remind everybody that when you look at in your Congress bag, you will find the epilepsy mission. It's there for you, for your review and kind of consultation. So please read it. It has been prepared by the epiXchange consortium. And we would very much like to hear your comments. So send those to me and Janet Mifsud. So our email addresses are there. So we will do our best to adapt the final version of the document accordingly. So this is very important for the whole epilepsy community. So please take a look at the draft of the epilepsy mission. Okay, so now let's start with the first thematic session, and that is on biomarkers. And as you noticed, practically every consortium is working with epilepsy biomarkers. So that we all are on the same page. So first, let's look at what is the kind of an official or agreed definition of the biomarkers. And this is based on the best document. That is the collaboration of NIH and FDA. It's a PDF file available in the web. This is a document that has made a very good attempt to standardize the nomenclature and definitions, because it has been kind of soft over the years. So I would very much recommend that to everybody if you haven't yet looked at it. So according to that, a biomarker is a characteristic that is measured as an indicator of normal biological processes, pathogenic processes, or response to an exposure or intervention, including therapeutic interventions. It has different modalities. They can be molecular, histologic, radiographic, or physiologic. So when we now look at the different biomarker categories, so as was already indicated in one presentation earlier, we have 7 different categories: susceptibility or risk biomarkers, diagnostic, monitoring, prognostic, predictive, pharmacodynamic or response, and safety biomarkers. And you can see here the reviews from EpiTarget and others. And this slide also very clearly shows the gap that we have in the epilepsy field. I have put on bold those categories to which there have been some attempts to find biomarkers. But as you can see, there are 3 categories in which we don't have any published biomarkers which come with a statistic showing sensitivity and specificity. So regarding filling up the gaps, here are the presentations or presenters and which biomarker categories these presentations actually address. The next speaker will be Professor Rosenov, and he comes from EpimiRNA, and his title is MicroRNA as Biomarker of Epilepsy and Status Epilepticus.
Thanks, Asla, for the kind introduction. I will now try to cover what we found in the EpimiRNA regarding biomarkers, and I'll see whether I can forward this. I think that's the main bottleneck and challenge today. Very good. So why should we choose to use microRNAs as biomarkers for epilepsy? Well, they're stable in different bodily fluids. They are easily detectable and quantifiable by methods such as PCR. And they're pathophysiologically relevant, which makes them more interesting. They are brain-enriched or sometimes brain-specific, and they are cell-type specific at times. They're closely related to the epileptogenic abnormality, and that allows for the identification of therapeutic targets and also opens the option of therapeutic monitoring. as a response biomarker. They're measurable in the POCT setting. You see a device here that was developed in Dublin, which allows to measure, for instance, microRNA 134 within 1.5 hours in the bedside setting. And there's also some standardization going on right now. This is a paper by Asla and colleagues looking at animal blood research. And standardization is very important because it's so complex, as has been mentioned before. So what were the sub-projects that we followed was one, plasma microRNA as a diagnostic biomarker in mesotemporal lobe epilepsy and seizures. Then we looked at CSF microRNA as a diagnostic biomarker for mesotemporal lobe epilepsy again and status epilepticus. We looked a little bit, and I'm not going to go into this in much detail, as microRNA as a potential treatment response biomarker for therapeutic brain stimulation. This was experimental. And I will not go into the extensive biofluid analysis in 3 rodent models that we also undertook. So this is some results from the plasma microRNA approach. See, in Marburg and Dublin, we sampled initially 32 controls, 32 specimens from patients with temporal lobe epilepsy as a baseline sample, and then after a seizure. We had a dual platform approach looking at open array platform with 750 microRNAs assessed and then RNA sequencing. The latter shows us these results, which are reproducible between the 2 cohorts. For IP protection purposes, I'm not able to show you the actual numbers, but it was some 16 dysregulated microRNAs, and they could be confirmed in phase 2. microRNA 122 was taken out, and the other 15 were mainly brain-enriched, and 80% of those are found also in EpimiRNA base, which is an indication that they're actually involved in epilepsy or epileptogenesis, as proven by other publications. Here you find 12 out of 15 were confirmed in the confirmatory phase of the study, and I just show you the 3 most significant ones. You see here one where the control as compared to the epilepsy baseline levels is significantly different. See the ROC curve analysis here, which shows that it has quite nice area under the curves with approaching 0.9. This would be for epilepsy versus controls, whereas here you have the main differences between before epilepsy as a baseline, before a seizure, and then after a seizure. So this would be seizure biomarkers with, again, quite nice area under the curves regarding the ROC analyses. And then we looked at this in a big validation cohort of some 263 patients with about a little bit more than 300 samples, including 102 patients with temporal lobe epilepsy, 10 with frontal lobe epilepsy, idiopathic generalized epilepsy, non-epileptic attack disorder, also called psychogenic seizures, status, and 110 controls. And again, we have significant differences regarding these 3 microRNAs between controls and IGE, idiopathic generalized epilepsy, but mainly as compared to the temporal lobe epilepsy cases. Looking at CSF, microRNA in the CSF. Why should you use CSF? Well, there's obvious advantages. You have a closer contact to the brain tissue. What you will find is probably brain-expressed microRNA mostly or only. There are disadvantages. It's obviously more invasive. It's not done on a routine basis in clinical practice, and the microRNA concentration is somewhat lower as compared to the plasma samples. We had samples from several universities, including Erlangen, Magdeburg, Marburg, and Dublin, and we looked at temporal lobe epilepsy and status epilepticus and at controls. In the discovery phase, the controls were mainly headache patients. We had 3 times 15 samples here, and in the validation phase, CSF from patients with other neurological diseases was included, mostly Alzheimer's disease and MS. And here you see the 3 most significant ones again, this time with the numbers because this is published already. And you see that the 19B3P was most useful to discern temporal lobe epilepsy from the 3 other conditions compared to 21-5P and 451A, who were most useful to discern status epilepticus from the 3 other conditions, and you see the related p-values here. Area under the curves, again, approaching 0.8 or 0.9 for these 3 single ones and for these comparisons. And there we looked at whether the combination of the 3 might increase our area under the curve, specificity and sensitivity, which was the case for temporal lobe epilepsy versus controls, where we had lower values initially, and then by combining the 3, you could come to higher sensitivities and specificities. So that's an approach that might be taken in the future. The next sub-project was looking, it is presented in a poster here, so I don't need to go into this in much detail. We looked at different stimulation paradigms in a rat model of peripheral path-based stimulation, temporal lobe epilepsy, and we found that the low-frequency stimulation of the ventral hippocampal commissure was the most effective way to stimulate in order to prevent epilepsy or prolong epileptogenesis. This had anti-epileptogenic effects, so it took longer, significantly longer, until seizures first developed, but we could not suppress the development of seizures. You see that over time, usually this microRNA goes up and then goes significantly up at day 97, which can be prohibited or suppressed by the ventral hippocampal commissure stimulation, where the levels remain the same, and this increase is inhibited. And the number of other microRNAs were also differentially expressed with that stimulation. So what are the achievements, not only by microRNA, but this is certainly also achieved by other people? MicroRNA is established as a new potential biomarker of temporal lobe epilepsy and status epilepticus and seizures. There are standards for measuring, which we have now been establishing in the clinical setting. Plasma is better than serum. Argo pull down, so exosomal microRNA arrive at different, thank you very much, arrive at different results. There's a novel way for point of care quantification has been established, and I mentioned this just a second ago. So what are the challenges and bottlenecks? I chose to build this up, which is a big problem in the current setting here, but I will try to go through it as fast as possible. So mainly we have now diagnostic microRNA used as a diagnostic biomarker in a few specific epilepsy syndromes, such as mesotemporal lobe epilepsy. It doesn't seem to want to go ahead. Oh yeah, it's still quite time-consuming and expensive to measure microRNA. That are the main bottlenecks I would like to address today. And the steps forward are obviously related. We need much more sampling in different specific epilepsy syndromes. We need to come up with standardization and better and faster ways to measure microRNA. Yeah, and I don't remember the other 2 ones, but I think we're running out of time. Thank you very much.
Let's move on. The next speaker comes from EpiStop, Professor Lagay. And he's going to talk about EEG changes as a biomarker for emerging epilepsy in TSC.
Thank you very much. I will focus on EEG as a marker, I would not say a biomarker, for following epilepsy and the value of EEG in predicting and for prognosis in epilepsy. I have more questions and answers, so I will start with a couple of assumptions. This is a very simplistic overview of the ideas we had before EpiStop started, and these are really assumptions. Okay, first of all, and you might agree on this, in some way, EEG changes do reflect epileptogenic processes. I'm not sure that if that's true, but that's what people assume. And then the second one is, the more EEG changes you see, the more likely that a patient or a child will develop eventually epilepsy. We also know for sure that if epilepsy starts very early on, and if it's really refractory, that prognosis will be very bad. And I'm talking about very young children below the age of 1 or 2 years. So the idea is, if we could interfere with epileptogenesis before the start of the clinical seizures, perhaps we can make the outcome much better. But immediately, and this might also be my conclusions already, all of these ideas might be completely wrong, because we know in clinical practice that many children or adults have spikes on their EEG and never develop epilepsy. If you read papers on ADHD, up to 25% of children with ADHD do have spikes, they never develop epilepsy. So it's not, if you use the terminology of the best paper, not a diagnostic or a predictive biomarker EEG. First of all, we do see children, for instance, with absence epilepsy, where you control the absences completely. Everybody is very happy, but if you run an EEG, you will still see generalized spike and wave. So you cannot call it a real nice response biomarker, the EEG. And worse EEG findings, that means a lot of spikes or exactly, for instance, during the night, it's not always correlated with a worse EEG. So in some animal models, this hypothesis of epileptogenesis being followed by EEG changes is actually true. So if you have a traumatic brain injury, first you see the spikes, and then eventually later on, only the seizures will come forward, and the more spikes you see, the more seizures. So this is one model, just one model. In humans, there are a few studies, and actually not too many studies, showing, and this is the field we are entering with EpiStop, that you could, before the appearance of tonic spasms, epileptic spasms, the EEG might, before that, show already focal spikes, the gray, or fast rhythms, or even hypsarrhythmia, and only then afterwards you will see the occurrence of epileptic spikes. So this shows that there might be some prediction of the EEG findings towards the occurrence of epileptic spikes. But these three cases are completely different. They follow the EEGs in these children, and they have, like, for a couple of months or almost 1 year, focal spikes on their EEG, fast rhythms, and they never develop epilepsy in the first 2 years of life. Now, let's turn to tuberous sclerosis. As you all know, this is a multisystem disorder, genetic disorder with a mutation in TSC1 and TSC2, upregulation of the mTOR pathways. And we know that many of these children will have seizures, and especially, as Sergius already mentioned, in the first year of life, about 70% of these children will develop epilepsy, and that the seizure types that they will show are focal seizures or infantile spasms. So this is a classical picture, and most of these children with onset of epilepsy before the age of 1 year really do not very well at the developmental field. And then this paper came along, and this is the paper of Sergius, small paper, but very influential, so published in 2011. They would follow children diagnosed with tuberous sclerosis at birth sequentially with EEGs every 4 weeks or every 6 weeks, and whenever there would be multifocal spikes on the EEG, they would start vigabatrin. And so they compared that group of what they call preventive treatment with the standard of care treatment. So small groups, 14 children, but if you look at the differences, for instance, that seizure-free patient at the age of 2 years in the preventive group, 92 was seizure-free versus 35% in the standard of care group. Standard of care means that you would only treat infantile spasms or focal seizures whenever they occur clinically. The IQ, which is actually even more important, 92 in the preventive group and 68 in the standard group. So this very small paper showed that preventive treatment based on treating the EEG and not the clinical seizures might be of any benefit for these children. There's another paper, prospective paper, only one of the few papers on this, following from the States, following again tuberous sclerosis children prospectively for more than 12 months with regular video EEGs, and again they showed that about 70% of these children do develop epilepsy. They could show that the first EEG abnormalities appear around 4 months. In EpiStop, we see it's much earlier. Onset of epilepsy, there was a delay of about 2 months. And they showed that they predicted that if you do see epileptiform discharge on the EEG, all of these children will develop eventually clinical seizures. So this would be a prognostic biomarker. If you don't have any epilepsy on the EEG, you don't know what will happen. Now, if you look in more detail to that paper, so there are children with normal EEG and no seizures. There are still children with normal EEG, and at some point they would develop focal seizures. And this is the largest group. They would have first a normal EEG, then abnormal EEG, and then epileptic spasms. Now, in EpiStop, we're still trying to replicate the data of Poland. So this is a randomized controlled trial, as Sergius explained to you, and we are still analyzing all the EEGs. We have about 2,000 EEGs to analyze, and so we have a randomization study. So we ask the parents to sign up for the randomization study, or they will have standard of care treatment, meaning you follow the children, and whenever they present with infantile spasm or focal seizure, you would treat them with vigabatrin, or you base your randomization on abnormal EEG. So if the EEG is abnormal, before the onset of the clinical seizure, you would start with vigabatrin. The primary outcome is development at the age of 2 years and the occurrence or the delay of clinical epilepsy. We cannot show too many results yet because we are still analyzing and collecting developmental data, but a couple of issues that are important if you talk about biomarkers. Actually, at the age of 2 months in our cohort, about 100 children, as you heard, 92% do have spikes. So what's the value of spikes? Almost 100% of these children with tuberous sclerosis will develop during the first year of life spikes on the EEG. And if you look at the sort of EEG spikes, you can look at focal spikes or multifocal spikes. A means very few spikes, and 4 means a lot of spikes, more than 50% of the time. So the first abnormal EEG in our cohort could already be a very disturbed multifocal epileptic spikes with more than 50% of the time. So you can start with a very few spikes, or you can start with actually many spikes. Now then, of course, we look at the occurrence of seizures, and in 70 or 80% of these children, we do see seizures. In contrast to the American study, if you have a normal EEG, we still have, of course, also 2 children with seizures. If you have a minimally disturbed EEG, score A, you really don't know what will happen. If you have a score C, which is multifocal spikes on the EEG, you see that the majority of these children will develop clinical seizures, but this is not statistically significant. So to conclude, I think I offered you some challenges already. So what is the value of a spike on an EEG? I really don't know anymore. And I think we should look not only at spikes. These are not sensitive enough. We really have to look into more advanced signal processing, background analysis, coherence analysis, correlation analysis. This is the way to go forward. And of course, I think in this world of tuberous sclerosis, we have to combine, as already said by one of the other speakers, we have to combine markers, biomarkers. We have to combine it with imaging, molecular data, and genetic data, and only then probably we will predict the onset of epilepsy in some of these children with tuberous sclerosis. Thank you very much.
So thank you, and thanks to the speakers for remaining pretty much in time. Unfortunately, the introductory session was longer than we expected, so we don't have much time for questions. As you've seen, the idea, the scheme will be to have flash presentations and then a general discussion that will inform the paper that we want to produce. So are there questions at the end of this session? Piero?
So the methylation patterns that were being picked up in the focal cortical dysplasias, do they correlate with any somatic mutations in those individuals? Like a proportion of those cases can be explained by somatic mutations, whether there's a correlation between the methylation pattern and a genetic diagnosis?
Okay. We didn't do exome sequencing or whole genome sequencing on these patients. We did sequence a panel of 53 genes from the mTOR pathway and other genes which are related to brain development to at least exclude any mutations here. We have 2 patients in the cohort which show mutation in DEPTC5 and ACT1, but beyond that, we didn't see anything. So basically, from our understanding, it's still the picture that you have a smaller group of patients which show a mutation, but the others are unclear, and this is why we think that the study of the DNA methylation helps us maybe get some more insight.
Other questions?
Well, maybe I have a question for the panel and also for the coordinators. When we organized this session, we left the topics completely open. And at the end, we ended up with essentially 4 presentations that are on molecular or genetic biomarkers and 1 on EEG, and no presentation at all on imaging. So since I know that imaging studies for identification of biomarkers were ongoing in the projects, I was wondering if that means that those attempts failed, or is there any information on that from the speakers or from the coordinators of the projects? Anna Maria?
We have done in EpiTarget some studies with MR spectroscopy by measuring the myoinositol levels that reflects the astrocytic activation, and using the same models that have been shown before, Teresa Ravizza found out that indeed you can predict the development of the disease by measuring the myoinositol levels that remain high in the animals developing epilepsy, while they have an early increase and then go back to baseline levels in the animals that, being exposed to status epilepticus, do not develop the disease. And she also found out that some of the behavioral comorbidities, cognitive deficits, measured in the Morris maze, were also predictive of epilepsy development, because in many of the models that we are using, these comorbidities occur before the spontaneous seizures. So I think that imaging is, I personally think it's a good opportunity for finding and looking for biomarkers.
And behavior as well.
Hello, my name is Floor Janssen. I'm a work package leader of the imaging in EpiStop. And what we tend to do is look at the different MRI characteristics in comparison to patients who develop epilepsy and those who, in relation with the cognitive outcome, in relation to work package 7 of Paolo Curatolo. We look at the different type of lesions in the tuberous sclerosis, so the tuberous but FCD-like type lesions, the SEN-SIGA, but we do not expect those to be related to epilepsy. We look at the lesion load, so we do segmentations of all the lesions, and we look at DTI characteristics. Unfortunately, a few centers were able to form arterial spin labeling echo, ASL techniques, but it's still work in progress. The one thing we can say now is that indeed the children have a higher lesion load in the beginning, so looking at the proportion of the lesions in the total brain volume, we see that those are more prone to have seizures, but we do not have the results yet because we do not have the 2-year outcome for all the patients, but we would love to present that in another occasion. It's something we will still, we will do, but we haven't done it at this moment. Yeah.
Also member of EpiStop. We did this, and there is a part on just a few patients where we compared the same day we did the MRI with the arterial spin labeling with concomitant EEG recordings, and this can be really a very promising, adding to what Floor was saying, doing this concomitant EEGs. But this is, as you all know, huge time consuming, very difficult in patients who had mental delay, hyperactivity, because we need them not to be on general anesthesia to do this.